We've just published a paper in Current Biology, and it's a remarkable piece of work. It's a collective effort. It's I think, a uniquely Cornell story. It is the first time, it's the first report of the neurophysiological analysis of vision in a jumping spider. Let me introduce the members of the team. So to my left, we have Gil Menda, who is the electrophysiologist on the team.
This project was when I was a grad student. Currently, I'm a postdoc in the lab. I'm the neurophysiologist of the team. So I did the recording with the jumping spiders, solved the problem of recording with jumping spiders. Since they are a little bit problematic, they are carrying high pressure in their body, and whenever you poke them, they explode and die. And what I did here is actually solve that method and bring all the data to the team, and then the team took the data and create all of the analysis.
To my immediate right is James Golden, who is doing his PhD thesis is actually on humans.
I'm a PhD student in the Department of Psychology. I joined the Hoy Lab a few years ago. I met Gil when he TA'd a neurophysiology course that I took. And when we started with the spiders, I was helping to build the movie stimuli that we use. And I also help with the analysis. I do the spike sorting, and I help build the figures.
To James's right is Paul Shambo, who is doing his PhD jointly in my lab, and Rob Raguso, our chairman.
I'm a PhD student here in neurobiology and behavior. I'm part of the Hoy Lab. And as far as this project goes, I suppose I'm sort of the resident arachnologist. And I also did a little bit of whatever was needed, a little bit of programming here and there for building stimuli and analyzing data.
To Paul's right, we have Eyal Nitzani, who has so many titles and affiliations that he'll have to speak for himself.
I'm a PhD student in the program of computational biology and medicine, the CBM program of Cornell, which is used to call the Tri-I program, which includes the Memorial Sloan Kettering Institute in the city, Weill Cornell Medical College also in the city, they're just across each other, and Cornell University. I'm actually doing my PhD here. I have two supervisors. One is here, the other one is in the medical college, and I'm in the department here. I'm in the biological statistics and computational biology, and in the department in Weill is the Brain and Mind Research Institute. So my main contribution to this project was about the analytical stuff, statistics, and also we brought some very unique mathematical stimuli that help us expose something about the integration of different kinds of inputs. Let me show you some of the stimulus that we used in order to expose this very unique phenomena about the spiders. So here we have it here, and this is actually, most of you would probably look at this, and this is kind of a white noise. It's moving to the right and it's moving to the left, and then you see some other kinds of things. And so using this stimuli, this is a very unique mathematical stimuli that each pixel is black and white and changing in different frame rate, and we can use this stimuli in order to expose the interactions between the eyes.
Our vision group are investigating the nerve basis of the vision of jumping spiders for the first time. We are working with Philippus adex. This is a jumping spider that mainly in the northeast of the United States. So I put the spider in the refrigerator for a few minutes just to slow her down. Take the 3D holder that we have designed. Okay, once the holder is set like that, the next step is actually to wax the chelicera's. It's very crucial that there will be no movement artifact once we are recording. A template here, the holder. I can lock it here on the side with wax. So the brain is located between the two eyes, the two rear eyes, and I will poke around this area over here. So the trick is to poke the spider in the right location, because I do only one hole, try to do one hole because she's eye pressurized. Very gently I'm poking the cuticle until I open the surface of it. Since I really want to try to make a very, very small hole. Okay, so we are using a extra small electrode. It's a 4 mega ohm tungsten electrode. And so the first step is just to lower the electrode to the right location. So the receptive field is in this area on the left. Can you see the laser? Okay. So what you can see on the screen is the appearance of the fly, and once the fly in the right location in the receptive field of the spider, and the spider recognizes it, you can hear this very, very strong response of these several units. So we can see here several classes of neurons that are responding to the appearance of the fly. The spiders are unique since they deserted the web in their evolution, and they actually use their eyes and food to hunt their prey. They have their big eyes, the median's eyes, resolution is almost as a human resolution, a little bit lower. But since in our eyes we have the motion and the acuity in one eye, the spiders just separate the motion to the lateral eyes and the eye acuity with a moving retina to the anterior median eyes.
You've now seen how 4 very talented students who come from diverse backgrounds can pool their talents working as a team to crack one of the toughest problems in spider biology. Now, we've done so in jumping spiders, and of course, jumping spiders, if it's not obvious from what you've seen earlier, are very small. And it turns out that another group of scientists, or engineers and roboticists, are also interested in how very small animals do remarkable things. And these are roboticists or engineers. They are always looking for ways to miniaturize biosensors, and in this case, visual sensors. And it has not escaped our notice that jumping spiders, having evolved a unique set of eyes, one set outsourced for detecting motion and another for very fine vision, so fine scale that it's almost as good as our own eyes, this should interest the roboticists.